temperature. And I just read that, yeah, don't go high, just keep it kind of low. Get it more low. So easy, but no. People. So, okay. Hello, everyone. Welcome to today's botany seminar. I have the pleasure of introducing today's speaker. I'm Dave Williams, a professor in botany. These moments are bittersweet. We are seeing Abby Hoffman finish up her dissertation work and go off to greener pastures in Alaska. These are bittersweet moments. I mean, it's great that Abby's gotten to this point where she's just a couple of steps away of earning her doctorate. But of course, it's unfortunate for all of us who will be losing a colleague moving away to another place in remote Alaska to better prepare the Alaskans for the future. So Abby has a lot of wonderful qualities as a graduate student, and I'm going to just dote on her for a little bit here. She's an ideal graduate student. Abby's been absolutely wonderful. She is tenacious. She is super charged about her research. She has taken ownership of the work that you're going to see today in the presentation at a really high level. She's creative. She's a fantastic writer. What else can I say? She works really hard. Maybe I already said that. She's the ideal graduate student. She's made it really easy for me over the last 4 years to be her mentor and friend. Abby came to the University of Wyoming from Duke University, where she finished up her bachelor's degree there. She moved out west and started a project with Shannon Albeck and I, and did a master's degree doing a really cool project in the Greater Yellowstone looking at atmospheric nitrogen deposition. She decided to stay on, good for us, and join the microbial ecology project, the EBSCO program, and has done some amazing work there. She's here to present her dissertation work about microbial ecology and biogeochemistry in annual snowpack, a very uncharted territory. And she's learned a lot. She's taught me a lot, and hopefully you'll learn a lot about this very fascinating topic. So I'm happy to present to you, and let's all welcome Abby Hoffman, who's presenting her dissertation work.
Thanks, Dave. That was probably a too kind introduction. But thank you all for being here today in person and also online. It's so great to see everyone. And today I'll be giving my dissertation talk titled Patterns of Microbial Assemblages and Biogeochemical Activity in Mountains. So I'll start off with an outline of the talk and actually give you the key takeaways here. So I'll start off with some background on microbes in snow and talk a bit about how these snow ecosystems are changing and why they matter. And then I'll talk about the first 3 chapters of my dissertation. In chapter 1, we found that microbial enzyme activity is high in seasonal snow. In chapter 2, we found that bacterial communities change rapidly in late spring snow. And in chapter 3, we did a nutrient and carbon addition experiment. And we found that bacterial communities, but not activity, responded to additions of carbon and nutrients in seasonal snow. In addition, I had a 4th chapter in my dissertation that investigated perennial snowfields as a source of microbes, carbon, and nutrients in alpine ecosystems. But I'm not going to have time to talk about this today, other than to just show this beautiful picture of Felix helping out with some field work up in northern Wyoming. So mountain snow provides many ecosystem services. Nearly half the world's populations get their drinking water from mountain snow. And in addition to water, mountain snow affects sediment fluxes and erosion, as well as nutrient fluxes to downstream ecosystems. So mountain snow is actually important in downstream freshwater biogeochemistry and aquatic ecosystems. And in addition, mountain snow is important for mining and hydropower, agriculture, and also for recreation. However, while mountain snow is largely recognized to store water and nutrients, we know nearly nothing about how microbial communities in seasonal snow might actually alter melt chemistry and alter inputs to downstream ecosystems. And this is potentially important because different forms of carbon or nutrients might affect downstream ecosystems differently. For example, more labile or recalcitrant carbon inputs to downstream ecosystems could alter impacts on soil microbes or aquatic chemistry. And in addition, different forms of nitrogen can impact ecosystems differently. For example, ammonium ions are more likely to be retained in soil and taken up by plants, whereas nitrate ions are more likely to be leached out of the soil and end up in fresh water and alter water quality and downstream ecosystems. And globally and across the Western United States, snowpack is declining. So this figure is just showing trends in snow water equivalent to the amount of water stored in snowpack over the last roughly 60 years. And losses in snowpack over this time period have resulted in roughly a 15 to 30% loss of snow water equivalent. And in addition just to loss of snowpack, we're also expecting to see changes in snowmelt patterns. So this figure on the top, we have patterns of higher melt. In the middle, we have patterns of moderate melt. And on the bottom here, we have patterns of low melt. And this is shown under two different scenarios. So our dotted lines are our control or our current scenario, and our solid lines are under projected global warming. And so the first thing that you can see is that all types of melt are shifting earlier in the year. But what's really interesting here is that we actually get lower frequencies of high and moderate melt, and we get more extended periods of low melt. You can see down here on the bottom, and that's because we're getting more warmer days earlier in the year, such as today. And but there's lower solar radiation early in the year, so melt actually doesn't occur as quickly early in the winter. And so potentially this extended period of low melt could be really influential on potential microbial activity. In addition to changes snow accumulation and melt patterns, we're also seeing alterations in global nutrient cycles. And so over the 20th century, we saw increased nitrogen deposition. That's what this top figure is showing. And that's largely due to nitrogen can volatilize off of animal waste and fertilizers, and fossil fuel combustion also produces atmospheric nitrogen, in addition to natural sources of atmospheric nitrogen. And while these rates of nitrogen deposition have largely leveled off in the 21st century due to increasing regulation, we still see elevated nitrogen deposition over historical levels. Additionally, dust deposition is increasing. And while we do get natural dust deposition, just due to winds in the desert, basically, dust deposition is basically a function of wind speeds and land cover. And so all these different land cover changes are contributing to increasing dust layers in the snowpack. So you can think of land clearing due to agriculture and development, droughts and fires are all contributing to increasing dust. And down there on the bottom right, you can see some really distinct dust layers in the snow. And these actually increase melting, and they also provide potential resources for biological activity in the snow. Additionally, over the past 20 or 30 years, scientists who study snow and ice have started to argue for the cryosphere as a biome. Perennial snow covers large areas in the Arctic and the Antarctic, as well as in areas in the mountains covered by glaciers. And they're largely not considered in the biosphere. And while they might not contain as obvious or diverse of plant and animal communities, they do contain quite a diversity of microorganisms, which are pictured here on the right. We have some snow algae on the top there, and then there's also a mite and a tardigrade, the springtail. And while the snow is definitely a challenging ecosystem, there's relatively low resource availability in comparison with other terrestrial ecosystems, high UV radiation, freezing temperatures, and low liquid water availability. Microorganisms have many adaptations to adapt and grow in these ecosystems. And so often we observe oligotrophic organisms that are slow growing. They can utilize low resource availability, and they are good stress tolerators. And that's in contrast with copiotrophs, which we're going to talk a little bit more about later, which grow quickly and grow best in high resource environments. And in addition, many of these microbes produce extracellular polymeric substances, or EPS, and that's what's pictured over here on the right. On the top figure there, the pink is individual cells, and then they're surrounded by that blue film, and those are EPS, and they help to stabilize microbial communities. They're also pretty prevalent in biofilms. And then on the bottom there, that's zooming out just a little bit, and you can see how prevalent these substances can be in snow, and they contribute to basically a range of different functions that make the snow more habitable for microbes. So they can act as cryoprotectants, antidesiccants. They can help cells to maintain osmotic potential between their cell and the environment. And they can also contribute to sequestration of carbon and nutrients. And then in addition, many microbes have polar adapted proteins and enzymes that can actually be active at sub-zero temperatures. And globally, we know that snow is significantly contributes to global biogeochemical cycles. So on the top here, we have the amount of carbon that's stored in the Antarctic ice sheet, the Greenland ice sheet, and mountain glaciers. And then what's interesting here is on the bottom, we have the flux of carbon from these ecosystems. We actually see that the largest fluxes of carbon out of these snow and ice ecosystems comes from mountain glaciers because they're melting the most rapidly. And so this is kind of a general paradigm of carbon cycling in these large polar ecosystems. Most of what we know about snow and ice ecosystems come from these areas. And in the large interiors of ice sheets, we tend to see more sparse photosynthetic communities that are fixed in carbon, and these act as a carbon sink. You may have seen some recent high-profile studies that were covered like in the New York Times that talk about some feedbacks between large algal blooms that we're starting to see on the Arctic and Antarctic ice sheets that actually have positive feedback with melt. So we're getting increasing melt, increasing algal blooms, and it's very unclear how these ecosystems are going to change under future climate change. And then in addition, we have communities towards the edges of these ice sheets that have more marine or terrestrial inputs of organic matter and nutrients. And these seem to support more diverse, net heterotrophic groups of microbes. And so this is just to show you that even though we think of snow as relatively homogeneous, there are distinct environments that select for different groups of microbes. And microbial activity in glaciers do affect carbon and nitrogen cycling in downstream ecosystems. Studies have shown that organic matter that's microbially processed in glaciers is more available in downstream ecosystems and alters respiration rates. And in addition, these glaciers store nitrogen, and often glacier-fed lakes and streams have higher nitrogen concentrations and higher primary productivity, and often altered diatom communities. And so all of this work is based off of work done in perennial snow, but seasonal snow actually covers nearly 50% of the Northern Hemisphere's land masses annually. And so this kind of led me to think about, well, what if there are nutrient transformations occurring in seasonal snow? And when I began this research, there were a few studies that had basically sequenced microbes in seasonal snow. We know that they're there, but we had very little idea about whether microbes could be active in these environments. And there's actually reason to believe that microbial communities might be quite active. We get higher organic inputs than we see at the poles. There's higher nitrogen and dust deposition, as well as higher temperatures. But on the other hand, these are ephemeral ecosystems, and we don't really know how rapidly microbes can respond to the available resources. Okay, and so I showed this figure to give you an idea of how quickly the snowpack and snowpack resources can change when we kind of hit peak melt. So this is from a classic study done down in Colorado. And basically this shows that in the snowpack, ions melt out preferentially because they're not incorporated as part of the ice structure. So we get kind of these pulses of nutrients that drop off rather quickly, oftentimes within 10 to 20 days. And so, this is really kind of what was driving my research, was trying to understand how quickly microbes can respond to nutrient pulses and whether or not we see significant activity in snow. So now I'm going to talk about chapter 1, where we found that microbial enzyme activity was high in seasonal snow. For this chapter, we asked, what is the potential for microbes in seasonal snow to process organic matter and mineralize nitrogen and phosphorus? How does this compare to other freshwater ecosystems? So we compared our measurements to values from the literature for streams and lakes. And then what limits microbial activity in snow? And we hypothesized that we'd see higher microbial activity during snowmelt when we get those nutrient pulses, and that we might expect to see phosphorus limitation because there are relatively few phosphorus inputs in comparison with the organic litter that we get and the atmospheric nitrogen deposition. And in addition, we compared forest and meadow sites, and we expected that forest sites have higher litter inputs, so we might expect to see higher enzyme activity, while meadow sites have lower inputs, so we might expect to see lower enzyme activity. And for all of the studies, or all 3 chapters that I'm going to talk about today, we collected snow up in the eastern slopes of the Snowy Range, just west of town. That way. So for chapters 1 and 2, we collected at 3 different elevations from March to June. The lower elevation was by the Green Rock Trailhead, middle elevation was near Brooklyn Lake, and then the higher elevation was just below glacier lakes. These are all subalpine sites. And in chapter 1, I'm going to talk about the enzyme assays that we did. And then additionally, in chapter 2, I'm going to talk about, so at these same sites, we also collected samples to analyze DNA sequences. And so that's a picture of one of the filters that we extracted DNA from. And that's just to give you an idea of kind of the particulate matter in the snow and what these samples might have looked like. That's a late spring sample from a meadow site, so quite a bit of particulate matter there. And so to understand microbial activity, we measured extracellular enzyme analyses. And so basically microbes produce enzymes and they excrete them out into the environment, and they'll attach to an organic substrate and actually produce a product that microbes can incorporate into their cell. And I've listed the names of the enzymes that we measured, but I'm just going to refer to them as carbon or phosphorus or nitrogen acquiring enzymes for simplicity. And all of these enzymes catalyze terminal reactions, so they're actually producing a product that microbes can use. So for example, the phosphorus acquiring enzyme actually cleaves off a phosphate group, and that allows us to more directly connect snow resources with microbial activity. And so in addition to measuring absolute activity of these enzymes, we can also look at relative rates of activity to help understand limiting resources, because you can think of it sort of in an economic framework. If microbes have a limited amount of resources to put towards resource acquisition, they're going to put them towards the resource that is most limiting to their growth or activity. And so this is a large global data set, and on there is drawn a 1-to-1 line. And so generally when resources are abundant, the ratio of carbon to nitrogen to phosphorus acquiring enzymes converges on 1-to-1-to-1. But you can see that sometimes we fall off this line. So for example, if we look down here, phosphorus acquiring enzyme activity is much higher than carbon acquiring enzyme activity. And so we'd say that these samples are probably phosphorus limited. And so the way that I'm going to present my data is we actually calculated proportions because many of our samples have zeros, which make ratios very difficult. And so here we're going to have our carbon to phosphorus acquiring enzyme activity on the x-axis and our carbon to nitrogen acquiring proportion on the y-axis. So if these are equal to 0.5, that means that that enzyme activity is equal. And so this allows us to kind of divide up our space into these quadrants that are indicative of different resource limitation. And so you can think of this that if both of these proportions are high, close to 1, then carbon acquiring enzyme activity is high in comparison to both phosphorus and carbon, or phosphorus and nitrogen. So we call that more carbon limited, versus if one of them is high but the other is low, then that would mean that carbon is limiting in respect to phosphorus down here, but then nitrogen is actually limiting in respect to carbon. So we call that more nitrogen limited. So that's the way we're going to see these presented. Hopefully that helps. And so moving into some of the results. So this has been accepted for publication in Ecosphere, and hopefully this paper will be online any day now. And so we did find that enzyme activity was highest during melt, and it was higher in forest samples than in meadow samples. And if we take just those June samples where we had the highest rates and compare them with literature values for river and stream water and lake water, we see that these values are actually pretty high. And so this is pretty encouraging, and I think that this really warrants further investigation. It's important to note that compared with soils or sediments, those tend to have much higher rates of enzyme activity, although the units are a bit different that make it difficult to directly compare them. But this was pretty exciting to me. And next, if we look at our nutrient limitations, so here we have that carbon to phosphorus proportion on the bottom and our carbon to nitrogen acquiring enzyme proportion on the y-axis. Our snow samples are in green triangles and circles, and our lake waters and river and stream waters are in blue. And you can see that most of our snow samples fall in this phosphorus or carbon and phosphorus co-limited space. These samples that are way over here on the right tended to have very low enzyme activity, so it's possible that some kind of threshold amount of carbon or energy is required for enzyme production. But generally, these were phosphorus or carbon and phosphorus co-limited. In comparison with waters, which tended to be much more nitrogen and phosphorus co-limited. And so this could be a result of sampling at different temporal times that might cause this disconnect. But also this suggests that potentially nitrogen from the snow is important in shaping some of these aquatic ecosystems. And in fact, we do see that nitrogen inputs from glacial systems or from glaciers are important in glacial fed systems. Okay, so the conclusions from chapter 1 were that there was high enzyme activity in snow during late spring, and the snow tended to be phosphorus or carbon limited in comparison with lake and stream systems, which were more nitrogen and phosphorus. Moving on to chapter 2, where we found that bacterial communities changed rapidly in late spring snow. For this, we were really interested in to what extent microbial communities were able to change in seasonal snowpack. And for this, we expected that in early spring, snowpack is cold, it's dry, there's probably not a whole lot of activity. And so likely these communities are more deposition driven, and microbes are deposited in the snow in association with dust, but also microbes actually can act as ice nucleators, and so they can be deposited as part of the ice crystal. And we expected that these communities would probably be more likely to have more oligotrophic taxa that can tolerate more stress and grow under low resource conditions. And then later in spring, when the snow began to melt and we get warmer temperatures and nutrient pulses, we expected to see these communities to shift more towards resource or competition-driven communities. And we expected to see that reflected as more copiotrophic taxa in the snow. And so this is just showing you how snow chemistry changed over our sampling period. So as I showed you before, ions melt out preferentially in snow. So you can see here that ammonium nitrate and phosphate decrease across our melt period. But we actually get increasing concentrations of dissolved organic carbon pretty quickly in forest samples and to a lesser extent meadow samples. And so this is really what we thought might be driving some of these differences that we're seeing in snowpack, and why we thought Even as ions might melt out, we might expect to see more copiotrophic taxa. And so now this is our first look at our microbial data. So here we have alpha diversity of bacteria and fungi. So this is basically just showing you the total number of taxa in these different samples for both bacteria and fungi. And then our forest samples are in dark gray and our meadow samples are in light gray. And what you can see is that in June, our diversity decreased significantly in forest samples for both bacteria and fungi, but we didn't see any differences in the meadow samples. And in a couple of more recent studies in the Arctic, they've observed similar trends where they explain this that during the winter, when you get accumulation of microbes, basically that's just reflective of the atmosphere and it's a more diverse community of microbes. But then once snowmelt begins and microbes are actually able to grow, you get selection for microbes that are actually adapted to snow and able to grow in snow. And that's a smaller selection of the group of microbes that were just deposited there. And then if we look at kind of our broad categorizations of different groups of microbes, so we can categorize bacteria into these oligotroph or low resource, high stress groups versus copiotroph, high resource, fast growing groups, just based on their bacterial classes. Again, we see this trend where we get a significant change in the forest samples, but not in the meadow samples. And so these forest samples in June, they do reflect more copiotrophic taxa, which is indicative of a higher resource environment, which is kind of what we expected to see based off that different resource pulses in the snow. So in conclusion for chapter 2, we found that microbial communities in seasonal snow reflect variation in snowpack resource availability during melt. And we saw the largest changes in microbial communities occurred in June in forest snow. And so from these first 2 chapters, we started to develop this idea of how microbial activity and communities change during the winter and spring. So basically we thought that during winter and early spring, we get deposition of diverse microbes, but there's relatively low activity and microbes are limited by temperature and water availability. But then as the snow begins to melt later in spring, we get microbial growth and we start to select for maybe different groups of microbes in different environments. And so in forests, where there's more organic matter, we saw more copiotrophic communities, lower diversity, higher activity, and faster changes, whereas in these meadow samples, we saw there was less organic matter, lower dissolved organic carbon, there are more oligotrophic microbes, higher diversity, lower activity, and maybe these are changing a little bit slower. And overall, we also thought that these were maybe limited by phosphorus. And so I decided it'd be a great idea to test some carbon and phosphorus limitations with a nutrient addition experiment. So far, this work I've showed you was correlative, and there's so many things changing in the snowpack during snowmelt that it's difficult to understand what microbes we're really responding to. And so now I'll move on to chapter 3, where we found that bacterial communities, but not activity, responded to additions of carbon and nutrients in seasonal snow. And so for the experiment, in addition to measuring snow chemistry and enzyme activity and sequencing microbial communities, we also measured total phospholipid acids, or PLFAs. And PLFAs basically make up the cell membranes of microbes, so we can use them as a proxy for biomass. And this is a very useful complement to the DNA data because our DNA methods only allow us to understand relative abundances of different microbial groups. They don't tell us about whether the total abundance of microbes is increasing or decreasing or the same. And so here are just some fun pictures from the PLFA analyses. This really made me appreciate the organic chemistry classes I was forced to take. It's a pretty time-intensive process, but it's also like pretty interesting. Basically, we extract the organic material off of our filter with some fun organic solvents like chloroform and methanol. And then there's kind of a bunch of intermediate cleaning steps, and oftentimes samples look like this with some kind of cool looking different pigments and things. And then finally, we actually separated out the different lipid classes on these silica columns. And then we took what was hopefully our cleaned phospholipid sample. We ran it on the gas chromatograph, which gives us peaks for different biomarkers. And then we can add up all of our fungal and bacterial biomarkers to get an idea of biomass. Okay, so for chapter 3, we're interested in what limits microbial enzyme activity in melting snow and whether microbial taxa responded differently to nutrient additions in melting snowpack. So we did this nutrient addition experiment over 9 days in late May and early June. And we treated each plot every day because we knew that resources were melting out. And we tried to basically want to ensure we got a response. So every day we were adding 5 times the amount of resource that we measured in the snow the previous year. And we had 5 treatments. We had a control that was sprayed with water. And the snowpack is pretty saturated at this point, so we didn't expect water to affect these communities. But just to make sure at the end, we also grabbed some samples that were not treated with anything to make sure they weren't different than those water treatments. And then we also had a phosphorus treatment, a carbon and phosphorus treatment, and then a carbon, phosphorus, and nitrogen treatment as kind of a check that nitrogen wasn't important. And so we expected that across all treatments, we'd probably see an increase in microbial biomass and increase in enzyme activity because we expected this to be the most active period of microbial activity in seasonal snowpack. But in addition, we expected that, for example, under P limitation, we expected that adding phosphate would lead to some differences between our plots that were treated with phosphorus and those that weren't. And so we were hoping that if we're adding phosphate, eventually at some point we meet that limitation and we exceed it, and then we would expect to see a buildup of phosphate in these in snow as we've exceeded microbial demand for phosphate. And then in response to that, meeting that resource requirement, we'd expect to see phosphorus requiring enzyme activity to decline as microbes no longer needed to invest resources into acquiring phosphate because it was available in the snowpack. And then we expected that if we did add limited resources, then we should see faster increases in biomass in those treatments versus the other treatments. And so I'm going to start off showing the nitrogen data because just to kind of orient you to what's going on here. So we're showing on the x-axis, we have day of the experiment. And then on the y-axis, we have our nitrate concentration. And so on day 1 here, we collected that snow before we added any treatments. So you can see nitrate concentration is low. And then immediately, as we start adding nitrate, you can see that it builds up rapidly in the snow. And this is suggestive that microbes aren't really utilizing this resource. And we can combine that with our measured 2 nitrogen acquiring enzymes. And so here you can see that nitrogen acquiring enzyme activity is low throughout the experiment. And so that's additional evidence that microbes aren't spending many resources in order to acquire nitrogen, and it's probably not limiting in these ecosystems. However, when we look at our phosphate concentrations and our phosphorus acquiring enzyme activity, the story becomes puzzling. So we didn't see any differences between treatments for either phosphate concentrations or phosphorus requiring enzyme activity. And if you remember back, I showed you that plot where we had phosphate concentrations in the snow, and they should decline over the snow melt season, and they should be very low. And in fact, in all treatments, we see phosphate concentrations increase. Additionally, we see a similar trend with the dissolved organic carbon concentrations and with carbon acquiring enzyme activity. We see increasing dissolved organic carbon concentrations and increasing carbon acquiring enzyme activity across all treatments. And so I thought, well, the most obvious explanation for this would be that if by adding nutrients, we've just increased biomass in those plots, and then that extra biomass is utilizing resources, and so we're not seeing them in the snow. And so we can look at our PLFA biomass. And again, we do see increases in PLFA biomass, but it's not really different across treatments, although it does increase across the experiment. But this doesn't really account for why we don't see differences between our different treatments. And it suggests that maybe there are some pools or fluxes of carbon or phosphorus that we maybe have overlooked. And so here, this is kind of a simplified diagram showing some different fluxes with arrows and pools with circles. And so pools or fluxes that are colored in blue are things that we actually measured or that we know. So we know we added carbon and phosphate, and we expected it to alter the snow, phosphate, and dissolved organic carbon concentrations, which we didn't see. We also measured enzyme production and microbial biomass. We didn't really see differences between treatments there, but maybe there was some compensation from some of these other processes that explain why we don't see differences. Additionally, we haven't looked at our microbial community yet. So we also collected data to understand which microbes are here. And so we're going to look at that and see if that helps us to understand why we didn't see differences. And so here, this plot is basically showing you, so each point is a snow collection. And if points are closer together, that means that there was more overlap between those bacterial communities. If points were further away, that indicates that those communities were more different. And the different days are in different colors, and the different treatments are in different symbols. And so what's really interesting here that hopefully pops out to you is that the bacterial community composition changed pretty rapidly across these 9 days. And additionally, when we run our permutational analyses of variance, we see that both day and treatment had an impact on bacterial communities. And so this suggests that maybe there's some kind of compensations going on with the actual identity of different microbes that can utilize different resources. And so while the treatments aren't affecting kind of these broad measurements of ecosystem activity, maybe they're changing the microbes that are there that are then compensating for these different functions. And so we can look at this a little bit closer. So these are showing 2 phyla of bacteria that we measured, and that's kind of our highest classification. On the top here, we have proteobacteria, where our most relatively abundant phyla. And on the bottom, we have Bacteroidetes. And here you can see that in our carbon and phosphorus, our carbon and phosphorus and our carbon, phosphorus, and nitrogen treatments, we had higher relative abundances of proteobacteria versus our other treatments. And the other treatments tended to have higher relative abundances of Bacteroidetes in comparison with those carbon, phosphorus, and carbon, phosphorus, nitrogen treatments. And this might actually make some sense because we know that while these 2 phyla are actually pretty similar, they both would be classified as copiotrophic bacteria, so they can grow quickly and and they grow well in high resource environments. Proteobacteria tend to be very efficient at utilizing glucose for growth. And so might make sense that when we're adding glucose, we're actually selecting for those more. And then in the absence of adding glucose, that Bacteroidetes are known to be very effective at breaking down plant glycans. We know that a glycan is basically just a carbon molecule from plants. And so maybe in the absence of those large additions of glucose, we're selecting for microbes that can utilize the available resources there, which happen to be plant-derived carbons. And so this is super interesting because it suggests that even under altered resource availability, we're actually selecting for microbes that are best suited to those environments, and that those microbes are pretty resilient in maintaining ecosystem functions. And so to recap this, so across all our treatments, we did see an increase in biomass and an increase in enzyme activity. But we didn't see any real support or evidence against kind of broad scale carbon or phosphorus limitation on the overall community functions. And instead we observed that resource availability affected like individual microbial groups. And so rather than changing the kind of snow chemistry, found that additions of carbon and phosphate actually change our microbial activity or our microbial community. And that these different treatments selected for different microbial taxa, and that enzyme activities and biomass were resilient under these different resource availability. And so in conclusion, for chapter 3, we found that microbial community composition responded to additions of carbon and nutrients in seasonal snow, and taxa changes compensated to perform similar functions with different resource additions. And so with that, I already gave you my conclusions at the beginning, but we found that microbial enzyme activity was high in seasonal snow. Bacterial communities changed rapidly in late spring snow, and the bacterial communities, but not activity, responded to additions of carbon and nutrients in seasonal snow. And I think this is really exciting and really encouraging in the face of altered global nutrient cycling and altered ecosystems, that these bacterial communities may actually be quite resilient and quite a bit more complex than we expected to see in snow. So with that, I have a huge number of people to thank. Huge thanks to Dave, who just made these last 6 years great. I can't thank you enough for all of your encouragement and help and feedback. And I wouldn't be the scientist or the person I am today without you, so thank you. And then thanks to my committee, Shannon, Sarah, Linda, and Tice. I appreciate your time and feedback, and I'm excited to talk about this with you guys further. And then huge thanks to my lab group and a bunch of people in Pine EPSCoR who helped out with a ton of field work and lab work. It really is quite an effort to get out in the snow in winter. And I didn't show pictures of us digging pits down meters deep, but it's a lot of work, and I appreciate everyone who helped out. And then also huge thanks to Jessica and Greg and the labs here on campus who contributed to the production of a lot of this data. And then thanks to the Wyoming EPSCoR project for funding. And with that, I would like to take questions.
Those the taxonomic groups that you looked at for the bacteria are pretty broad, like as broad as you can get. I wondered if that was just because of the ability to resolve based on the data that you had, or if you looked at other finer scale things and whether there were any more with finer scale resolution, whether you could make any more conclusions about the
I definitely have finer scale data that we do see differences. It's so a lot of people will look at like the family level, and a lot of families are tied to functions, but we don't. So we group our sequences into these taxonomic groups, but we don't necessarily actually know their functions. We just know, okay, this family tends to have these genes, or so it's, you can start to extrapolate at lower levels for sure and speculate about functions at a finer scale level, but it becomes a problem of just the sheer amount of data, right? So you might have 100 families, and then you're trying to figure out relative abundances and which functions might be important. And it becomes very speculative very quickly. And so this is kind of a discussion that people are having in kind of microbial analyses right now, is whether we even should be like assigning functions to DNA sequences, or if we really should be measuring like proteins or RNA. But yeah, it's definitely possible to look at that. Time intensive is kind of why and usually you don't see these nice patterns at the high levels, so that's why I kind of pulled that out as like, this is actually really cool that we do see these differences at this high level.
Thanks.
So great talk. I really like how you complemented the microbial data with some of your more functional questions on a large ecological scale. So based on that, what is something in the future that you're really excited about pursuing in this field? Like, is there something throughout all of these experiments that you did that you're really excited to look more into?
Yeah, so this is something that I didn't do, but I think very interesting to know is a couple of studies in some of these glacier ecosystems have actually looked at the fluorescence of different carbon molecules, which can tell you about exactly what the carbon molecules are and how bioavailable they are, and whether or not, basically to what extent microbial processes are occurring. And I think that would be really useful to know, especially because, I mean, I showed you that we added glucose to these plots, and we don't actually know, are they actually utilizing the glucose? Is that why, are they just respiring it all off, and that's why we don't see differences? That's totally possible. Glucose might stimulate respiration, or like I kind of talked about with the Bacteroidetes, maybe utilizing more complex forms of carbon. That would help us to better understand some of those questions of what are the actual forms of these resources. And then that's also similar with the nitrate isotopes, where that allows us to better understand actual processing of some of these compounds by microbes.
Do you have some of these samples frozen at -80?
I think I do, but not enough to enact.
Talk to Mitch, because they can answer those exact questions.
Yeah. Well,
Chapter 5.
yeah, I'll take Daniel, and then we'll look in the chat on that.
Yeah, it's interesting that they were not nitrogen limited. So do you think it's always been the case, or do you think with increased nitrogen deposition that has changed over time?
That's a great question that I don't have any evidence to support either way, but I think we definitely see that atmospheric nitrogen deposition has altered nutrient limitations in terrestrial and aquatic ecosystems over the course of the 20th century. So I think it's likely that we have probably altered nutrient limitations. And as we have regulated nitrogen emissions more, we are seeing some shifts back to maybe more phosphorus limitation in some ecosystems too. So yeah, definitely a lot of alterations to these ecosystems that I think warrant a lot more research. Asking about why the ordinations kind of show that the early days grew more together. I think it's possible that in those early days, our treatments haven't affected our communities so much, and as we are adding more nutrients, and potentially this could be also because of maybe uneven application of nutrients, that we see more spread in those communities. But yeah, that's pretty speculative. I think just generally that these communities are changing pretty rapidly. I was surprised by how directional that change in the NMDS plot was. I thought that was pretty exciting. So.
You have one more at the bottom too.
Monitoring. Yeah, so this is a great point about microbes in snow. We don't know when they're deposited there, they might be dormant, but as soon as melting begins, they might very rapidly become active. And so we might not expect to see as strong of responses of biomass because of this, because the biomass is there, but we're just changing the conditions so it can be active. I think that definitely could affect biomass and composition. And I think that the data that I showed, along with some other data in Arctic ecosystems, shows that it's probably a combination of these two things, right? So you get deposition throughout the course of the winter, and maybe a lot of these microbes can't be active, but they are able to just survive in a dormant state. And then once melt begins, many of these microbes are able to be active and even grow.
Great talk. I don't know much about like nutrient dynamics and snowmelt, but if I recall correctly, like phosphates kind of stick right in soils and kind of binds to a bunch of like different substances it encounters. And I'm wondering, does that happen also in snowmelt or in snow where you have a lot of the dust particles? And could that explain like the pigmentation and some of those activities?
Yeah, definitely. So based on just my data from the first year that I collected snow, we saw low, very, very low phosphate concentrations throughout all of our samples. And so I kind of was expecting that because there's not as much organic matter or particulate as you see in soils, that it wasn't important. But actually, a study that came out late last year showed that in some of these algal blooms that we get, they actually are pretty good at retaining phosphate in the snow. And so I do wonder if that does contribute. Some of these late forest samples that we had a lot of particulate, and we saw those increasing phosphate concentrations. And so we might, there might be scenarios in the snow where we do get retention of phosphate because of that.
Okay, thank you. So the way I read that question, chat, was that they're saying that maybe there's an effect of predation on the microbes that you're measuring because you got bacteria eating. I mean, they're still small animals, but they're, I think they're preying on them. And perhaps you could have selective effects on your bacterial and fungal biomass from that predation.
Okay. Yeah, that's another great point. And I think there are a couple studies that have looked at grazing in cryoconates, are basically these pools of melted water on glaciers that basically accumulate around organic matter because the organic matter heats up the snow and it melts more. And if there have been a few studies that have looked at grazing in those ecosystems and found just some evidence that it might occur in the snowpack, or in seasonal snow, I should say. I don't know that I've seen any documentation of some of these higher level rotifers or grazers. So I'm not sure. I'm not sure I would necessarily expect those in snow. I don't know that they're being atmospherically deposited in snow, so it would be hard to think of what a source is, but I could be wrong. I don't know what kind of snow and ice environments that would be in. So it's possible for sure, but I think in seasonal snow, it's unlikely.
Hold on. Any more questions? Well, thanks everyone for coming, and thanks Abby for a great talk.
Abby, I'm guessing you did our